All right, so I want to talk to you just a little bit about gene expression and regulation. Just want to kind of highlight the stuff about the lac operon that you guys explored on your own already, and then give you just a brief introduction to what happens in eukaryotes, organisms with a true membrane-bound nucleus. And then I'm going to have another presentation that goes into more details on that. So the key concept is that gene expression is carefully regulated in all types of cells, both prokaryotic and eukaryotic, and it's much simpler in prokaryotic, as we're about to see. So remember, the operon is just a code for all the proteins that are going to be needed for a cellular function. And the operon includes a promoter, which is just the region where RNA polymerase binds in order to begin transcription, the operator, and one or more structural genes. Okay, so that could be one, two, three, so on and so forth. So just one or more structural genes. It really just depends on the operon that we're looking at. The operator is the section of DNA that basically switches the gene on or off, if you will. And by on or off, we simply mean on, it's being transcribed, off, it's not. And by it, I mean the gene. Operons are most common in prokaryotes. So in bacteria, genes are either on or off, like there's no in between. Significantly more complicated when we talk about eukaryotes in our next presentation. The lac operon was one of the first examples of gene regulation to be discovered, and you got that kind of historical information in the DNA from the beginning WebQuest. The lac operon has three genes that code for enzymes that break down lactose, one of the food supplies for bacteria, and it kind of just acts like a switch. So, what do we really mean when we say a gene is off or on? Well, if lactose isn't present, the gene is air quotes off, because you can see from the diagram that there's a repressor bound to the operator. And so that repressor is not going to allow RNA polymerase to transcribe those three genes for enzymes that digest lactose. And so we say the gene is off. We're not making those enzymes. There's no reason to, because lactose is not present in the environment. Well, if lactose is present, you can see from the diagram that lactose is going to bind to the repressor, which basically pops it off the operator. And then that's going to allow RNA polymerase to transcribe the information for the genes that digest the lactose. And so in that case, we say the gene is on. So on or off, no in between. Controlling transcription in eukaryotic cells is a lot more complicated. The genome is significantly longer. The genome will often bend. You can have enhancers, transcription factors. Eukaryotes can regulate genes at many different points, from the creation of the very first, like rough draft messenger RNA, all the way down to primary, secondary, tertiary, and quaternary folding, and all the spots in between. And I'm going to address that in more detail in my next presentation. Different sets of genes are expressed in different types of cells. So, like your eyeball cells will not create enzymes for digestion that your small intestine would. Transcription is controlled by regulatory DNA sequences and lots of protein factors. Most eukaryotes have a Tatabox promoter. That's just a section of the DNA code that's a bunch of T's and A's. And again, that just kind of tells the RNA polymerase kind of where this is going to begin. You need to start here, if you will. There are enhancer and silencers. So like these are just proteins that can either speed up or slow down the rate of transcription. Each gene has its own unique combination of regulatory sequences. So basically, it's just like too complex for PowerNote. So I'm going to address these things in a second presentation. And that is it. Thank you.